Okay, the next speaker is Professor Ali Mobasheri. His talk is about biomarkers of osteoarthritis, translating information from in vitro cellular systems to human tissues. Please.
Thank you, Mr. Chairman. Distinguished faculty, ladies and gentlemen, thank you to the organizers for inviting me to come and present at this prestigious conference. I'm jointly appointed at the moment between the University of Nottingham and the University of Bradford, and I'm also part of the visiting faculty here at King Abdulaziz University. In addition, I work with 3 other centers that I'd like to mention here, because the context is relevant to my presentation. These centres are funded by Arthritis Research UK, and the core focus of these is arthritis. Each centre focuses on different aspects of arthritis. One focuses on pain. We have a centre that focuses on sport, exercise, and the risk factors for osteoarthritis. And the one at the bottom is a joint centre between Nottingham and Birmingham, and this one is exclusively focused on the concept of musculoskeletal aging. In addition, I'm coordinator for D-Board, but I'll tell you about that in a moment. So, what I want to talk to you about today, I'm not going to show a lot of data. I'd like to give an overview of the area of osteoarthritis, biomarkers, and why we have dedicated so much of our time and effort in this area. Osteoarthritis is a major emerging health problem. It's one of the most common forms of arthritis, more common than rheumatoid arthritis, and a major cause of pain, inflammation, and loss of mobility in synovial joints. It's characterized by loss of articular cartilage and inflammation in the joint, and as you can see from the Does this work? Yeah, thank you. As you can see here, this is a relatively normal femoral head, and on the right-hand side we have an osteoarthritic femoral head with most of the cartilage worn away and the subchondral bone exposed. Now, the disease primarily affects load-bearing joints such as the hips and the knees, but other joints such as the hand, elbows, and shoulder can be affected as well. As I said earlier, it's a very common disease affecting approximately 10% of the population. No cure is currently available, and it's been designated as a priority disease area by the World Health Organization and various other funding agencies across the world. It's a major cause of disability. It's one of the top 5 healthcare issues in Europe and in the US, and is rising, and is going to have a major economic impact across the world. So I'd just like to summarize some of the risk factors. Of course, age is one of the key risk factors, and beyond the age of 60, many of us will have osteoarthritis in one or more of our joints. But obesity and having a BMI of greater than 30 is certainly one of the contributing factors. Damage, injury or trauma to the joint, excessive loads on a normal joint can be a major contributor, or joint instability, normal loads on a joint that has previously sustained an injury. Many of the speakers in this conference have been talking about genetics. There are a number of genes that are identified using GWAS approaches. A number of genes are potentially linked to the development of osteoarthritis, but there are potentially metabolic and endocrine contributing factors as well, and there's a link with diabetes also. Now, joint trauma is one of the key contributors to the development of osteoarthritis, and participation in sports that involve contact, collision, and excessive force on the joints are key risk factors. Sports such as rugby, football, even tennis, professional tennis, running are all potentially going to be high-risk sports. And what I've highlighted here is some of the mechanical factors that can contribute to osteoarthritis development in athletes. Injury itself, surgery which occurs after the injury, the muscle weakness after the recovery period, and the subsequent joint deformity, and what might happen in the joint which becomes unstable with repetitive joint loading. So obesity is also a major contributing factor, and this slide really highlights the sort of League of Nations when it comes to obesity. And it's a very interesting perspective looking at the correlation between the prevalence of obesity and the prevalence of osteoarthritis. Clearly, in countries such as the United States and the UK, there is a significant correlation between osteoarthritis and obesity. But it's not just excess weight. A lot of investigators in this area are beginning to realize that mechanical overload is one of the contributing factors, but it's the systemic inflammatory mediators and the pro-inflammatory cytokines produced by white adipose tissue that are making the major contribution to disease progression. So if you look at the main tissue that's affected in osteoarthritis, articular cartilage, this is what it looks like in the normal state. Smooth surface, healthy cellularity, and a single interface between the cartilage and the subchondral bone. And here on the right, you see disease. Fibrillated, fragmented articular cartilage, evidence of cells that are undergoing cloning and reparative processes, duplication of the tide mark, and significant vascular and neural changes in the subchondral bone. Here, in this situation, we have homeostasis between extracellular matrix synthesis and degradation, and in osteoarthritis, we have predominantly extracellular matrix degradation and cartilage collagen fragmentation. Also, the behavior of the cells changes. They become significantly aggravated and inflammatory in the osteoarthritic joint. When we look at the balance between cytokines and growth factors, the so-called catabolic and anabolic factors, there is a significant shift towards the ones that induce matrix degradation. And the ones that are involved in anabolic reactions are reduced in terms of abundance and activity. So that brings me to biomarkers. Why biomarkers? What are biomarkers, and why do we need them for osteoarthritis? Let me just give you a very brief definition of biomarker, which is a characteristic that's measured either as an indication of a normal physiological biological process or a pathogenic response to disease processes, or responses to drugs. And this definition has been around since 2001 and hasn't really changed significantly. There have been various refinements. Now, when it comes to osteoarthritis diagnosis, radiography is the gold standard. However, this gold standard is not adequate. By the time osteoarthritis is detected, there is significant joint damage and extracellular matrix changes in the cartilage. And the joint has gone potentially beyond the point of no repair. So there are significant inflammatory outcomes in a joint that exhibits changed joint space width. Here you see a normal joint on the left and an osteoarthritic joint on the right. And the change in the distance between the bones above and below is actually one of the key measures that's used in radiography. But this is an indirect measure of alterations in articular cartilage. It doesn't measure a dynamic process. It takes a snapshot at a particular moment in time. Any changes over time are subtle, small, and may occur in a small subset of patients, the so-called progressors. Radiographs can be poorly reproducible, and most importantly, the correlation with pain can be questionable. So in certain patients, the joints are extremely painful, and in others where there's significant cartilage loss, there is no pain. So when you think about biomarkers, they are very useful tools to assess the existence or the progression of different diseases. And I've borrowed this slide from one of my collaborators at Nordic Bioscience in Denmark to highlight some of the targets that they've developed and that they're working on in different disease areas. And the ones that are particularly interesting for us are the markers of the rheumatological markers and the markers associated with changes in muscle. Now, in osteoarthritis, biomarkers have a capacity to be able to detect changes early, before they've appeared on the radiograph. They can give us information about how the disease is progressing and predict the outcome of the disease. And when it comes to the different stages of osteoarthritis, it's not quite as complicated as rheumatoid arthritis. There are three distinct phases: a molecular phase, a pre-radiographic phase, and a clear recalcitrant radiographic phase. Now, in the molecular phase and the pre-radiographic phases, there is no pain. So the patient doesn't know that there is a disease process within the joint. And of course, by the time we get to the third stage, there have been significant and extensive changes in the articulating structures. And that presents us with a difficulty, because by that point, repair is going to be either difficult or not achievable. So we need a better gold standard. We need molecules, and we need diagnostic kits and assays that can give us early warning of joint degeneration and be able to give patients and their physicians an earlier and more targeted way to approach disease management. Also, biomarkers can facilitate drug discovery. If you look at the pharmaceutical industry at the present time, none of the major pharmaceutical drug companies have anything in their pipelines for osteoarthritis. Most of them have closed down their osteoarthritis research programs, and they find this a very difficult disease area. But we have to be optimistic, because genomics and the various other omics areas have the potential to reverse this situation and reinvigorate this area of research. Now, I've put this slide here just to highlight the fact that osteoarthritis and osteoporosis are closely linked. So in females over the age of 50, after menopause, the risk of osteoporosis also increases the risk of osteoarthritis. So some of the approaches that are going to be beneficial for osteoporosis are also going to be beneficial potentially for osteoarthritis. For example, bone conservation agents such as strontium ranelate. Some of the other things I've listed at the top are existing therapies. I mean, in fact, these are the only therapies that we currently have: analgesics and non-steroidal anti-inflammatory drugs. Other areas are questionable, or there might be weak evidence to support their use. Biological therapy is becoming quite popular in rheumatoid arthritis, and people are beginning to think that in very inflammatory situations in osteoarthritic patients, we might be able to use biological approaches to treating osteoarthritis, or facilitating cell-based, stem cell-based approaches. Another point to highlight and emphasize is the fact that biomarkers of arthritis can come from different tissue compartments. They can come from bone, from articular cartilage, or from the synovial membrane. So it's important to bear in mind where the tissue of origin actually is and what that molecule might be telling us. The other aspect I want to highlight is the fact that sometimes there might be a distinction between active and inactive proteins, or proteins that might be being degraded or those that might be turning over. And really, at the present time, a lot of the assays that are available are very poor. They don't tell us about proteins that are turning over. They might be telling us about proteins that might be degraded or processed in urine or blood as degradation products of extracellular matrix proteases. So we need to think about repair responses and degradation and put those biomarkers into context. But there are a number of important barriers that are currently preventing biomarkers reaching their full potential. The tissue source, the specificity, the lack of specificity rather, the underlying pathology, which can be multifaceted, and differences between the affected joints. An elbow joint and a knee joint are very different. Even the knee and the hip can present a different set of molecular markers. So, another important aspect is the fact that biomarkers, and many of the candidate biomarkers that are available, undergo significant variations in body fluids. And those variations can depend on renal function, on hepatic function, food intake, physical activity, and circadian rhythms and clocks. And we heard from some of the very nice presentations yesterday about the importance of taking those issues into consideration. This review from the Nordic Bioscience Group that we collaborate with highlight some of the other issues that are involved. When to do the assay, how to do the assay, variations in the antibodies that are available for those assays, and some of the very basic parameters that might affect assay results. The buffer, type of buffer used, the concentration of antibody. One kit might work differently in two different laboratories. So this area is fraught with many, many difficulties, conceptual difficulties. So we managed to convince the European Commission to fund our project, which aims exclusively at identifying new biomarkers and the ability to validate these in cohorts of patients. So we've called ourselves D-Board, and this is a project that was funded last October by the European Commission. They awarded us 6 million euros, and it's an omics-based approach. We have proteomics, lipidomics, and metabolomics, all focusing on articular cartilage and synovium from normal and osteoarthritic joints. And we have bioinformatics in there as a core to make sense of the data. So the aim of D-Board is to bring in academics and small companies to focus on identification of new biomarkers and developing new diagnostic tests that could identify disease and help with early disease diagnosis. Some of our participants are listed here. 3 of our participants are actually companies focusing on biomarker assay development validation. As I highlighted earlier, we've got various omics approaches with bioinformatics, all of them hopefully leading to the process of biomarker validation and assay development. Now, the title of my talk was taking information from in vitro models to patients. Now, in order to do that, we can use various types of in vitro models. We can use articular chondrocytes, synoviocytes, in culture, in co-culture, or explant tissues from animals. Or we can move to some of the animal models of osteoarthritis, ultimately to patients. But within our cohort, we have movement of information in this direction and in the reverse direction as well. And some of the things that we might have identified in animal models, we go back and revalidate in the in vitro models. And some of the studies that have identified molecules of interest in human studies are being revalidated in animal models to increase our confidence in the validity of those models. So the ultimate aim is to discover molecules, verify and validate them, develop assays and optimize those, and then take them to large cohorts of patients to allow us to achieve this very complex stage of biomarker qualification. And although we haven't made any specific promises to the European Commission, eventually develop and commercialize those products. Now, this is where biomarker research is placed in the context of community studies. Once you discover biomarkers, you need to validate them in community studies. And we're very pleased that we are able to collaborate with the Dutch cohorts in Rotterdam. They've been collecting samples from populations in Rotterdam since the end of the Second World War, and there's a specific emphasis on women's health There are a number of investigators investigating osteoporosis and osteoarthritis in women, especially post-menopause. So this is a very valuable tool. And also in Essex in the UK, we have the Chinkford study also focusing on osteoporosis and osteoarthritis. And some of the other cohorts that are being developed in these new centers, especially the sports cohorts, the footballers with joint injuries, are also going to be very useful to us. The one at the top left here is Euroteam, which is a team of academics focusing on rheumatoid arthritis biomarkers, and we have good links with them through the University of Birmingham. These are some of the companies that are involved in DBORD, Nordic Bioscience being the biggest one, and the one focusing exclusively on biomarkers in the areas of arthritis and cancer, and Artialis focuses on biomarkers of arthritis. So one of the programs that I'm leading is the proteomics program, and this focuses on proteomics. We use biological fluids, serum, plasma, urine, and synovial fluid. Synovial fluid is difficult to get, so we're finding it hard to build up a suitable biobank of these. But we're also using animal tissues, cartilage, bone, and synovial membrane, as well as the cells from these joint tissues, chondrocytes and synovial sites predominantly. So we can extract these, we can develop explant models or cellular models and look at what these cells secrete in response to pro-inflammatory and anti-inflammatory stimuli. And here's one of the models. We use, in this particular case, equine cartilage. We set up an explant model, and we set up a high-throughput proteomic system for identifying proteins that are released, and then we use antibody-based Western or immunoassays to validate. And just showing you some data from this platform. This platform allows you to look at matrix metalloproteases, because these are secreted proteins, and using the same methodology we've been able to show the effects of some of the existing anti-inflammatory molecules, NSAIDs, as well as some of the plant-derived anti-inflammatory compounds, such as curcumin and resveratrol. Professor Neyer gave a beautiful presentation about ion channels. We haven't forgotten about ion channels. We are very interested in cell surface markers of inflammatory change, and we are looking at alterations in ion channel expression and function in arthritis. And we do this work in collaboration with colleagues at the University of Liverpool. And we have a specific focus on potassium channels, because these channels are involved in metabolic regulation as well as regulating chondrocyte differentiation and proliferation. We're also interested in mechanisms that regulate glucose transport and metabolism. We're interested to find out what are the alterations in glucose transporters, as well as the enzymes that are responsible for glucose utilization and extracellular matrix glycosylation in cartilage. And we've already started to publish some of this work in collaboration with KAU here in Jeddah. I'd like to acknowledge my collaborators here at CGMR, the team of DBORD, our funding sources, particularly the European Commission, and thank you very much for your attention.